Hello students, welcome to EPG Pathshala. I am Indira Ghosh from JNU. Today we are going to talk on the module structure-based design and how interaction with active sites is going to happen under this paper called Computational Biology. Now, the learning objectives of this is the first step for structure biology design, and that is how we find the binding site, characterize the binding site, also how we define the interaction between ligand and protein. Then the search algorithm, that is how to find where the ligand can fit it or goes to be binding, so that we can go for the next step, that is docking the compound and searching for the scoring. I'm actually talking about the active site identification. It is very important to accurately identify the active site, and basically there are confusion about active site and binding site. Now, binding site is something where the substrate binds and activity or the property which we are trying to inhibit or accelerate happens. However, most of the cases, in case of mostly standard enzymes, or which we call that enzymes of normal types, you have, and following Michaelis-Menten equations, those enzymes normally are defined as their binding site and active site synonymously. That is, both are used exchangeably. However, when you look at the protein receptors and cases where there are transporters or more complex biological functions, those cases, many binding sites actually should perform as if it is an active site. Now, this is best to define as it is a functional part of the protein, which is recognized by the ligand, by the substrate, by the inhibitor, even by the cooperative binders. In case of cooperative binders, some other molecules like NAD or NADPH, etc. Now, the interaction site is a place where should be find out how the inhibitor or substrate or any of these, what we call commonly ligand, should bind. Very important to identify how big it is, which are the quality of this is. So not only we need to know the representation of the structure or the binding site or target site, which is with the help of crystal structures mostly, and in certain extent model and enema structures, one is able to find. One should also discriminate not only the whole structure but the area which is shown in the second middle of it, that connected points which connects only the active site. One should also find out the volume and surface area of the active site, which is shown on the extreme right slide. So, to go further, we must find some geometrical or some other methods which uses the coordinates of these proteins, maybe DNA and even maybe large carbohydrates, to identify where the ligand is appropriately can actually act, and the inhibitors should bind to the same place to stop the interaction. We discuss about how many methods are there. Actually, there are several different kind of methods. One is most commonly used is evolutionary identification methods. That is, from the sequence itself, one can find out how this identification of the active sites, which I'll talk last. However, other methods are mostly structure-based ones and using genetic algorithm or sometimes probe-based method. So if we classify all of them and find the residue, surface and volumes of these large cavities, sometimes even small cavities, we should be using three different groups, and we'll try to give a basic geometric logic of or scientific logic how that has been developed. Volumetric or geometric binding, we can categorize three of them. One is CAST-P, Pocket Finder and PASS. And more often PASS is used, but the other two are also very good softwares. Also, surface probe-based method, that is, identifying the active site is used by finding a probe and surface probe, that is, it searched in the surface of the molecule and try to find it. Liggsite and Subnet belongs to them. Also, there is a method evolutionary. As I said, I'll address that at the last. As told to you in previous slide, here I'm showing 4 different exclusive methods as shown in the picture. Now you can see the modules on the left hand side. The top has the ligand or ligand site protocol and how it is calculated. It is covered full thing protein by grid and then finding. The next to it is how the probe based method follows, and the bottom of the on the left hand side shows how the geometrically, almost analytically, one calculates the active site. And the last one, that is the bottom right, on the bottom right, shows the program called pass, which calculates the active sites by probes. So next slide we'll start taking each and every method and actually delivering and talking about the base method, the basic method, what we will go through it. Actually talks about the surface, that is cast P, which is called Computational Atlas of Surface, which is appropriate and has been hosted by us as a server. This uses very interesting method called Delaunay triangulation, which I'll come to next slides and all elaborate them, and an alpha complex for a shape measurement. This is a purely geometrical method. It also provides the surface accessible points and inaccessible points. That's the most important. And uses the Connolly or Richards methods, which is quite popular and known very well, and suggests openings as well as circumference of the openings, which is most important. Here I am elaborating how the triangulation are taken care in the figure shown behind. Triangulation is a technique by which you actually geometrically dissect the space. So consider a simplified, a hypothetical method where two-dimensional molecule formed by an atom disk. That is all of the same radius but atom disk, which is shown in the first A, figure A. Now if we take in the same figure A as it's shown, nail and figuratively hammered into the plane at each atom center, and a rubber band is stretched around the entire collection of nails, then the band will enclose something called convex hull of the molecule, which you can see in the figure A. Now this hull will contain all the atom centers within. Now convex hull of the disk, that is the centers in A, is the shape enclosed by the boundary, that is outer boundary of a polygon. By this way, you can check the figure B. Figure B in the shaded area is actually the surface. This can be triangulated, which is called tessellated with triangles, so that there is neither missing place nor there is any overlap of these triangles. This triangulation of convex hull, which is shown in B, figure B, the triangle tile of the shaded convex hull area, actually where the triangle tile that you can see now, all of the shaded convex hull area. This convex hull are the basic principle by which one can actually find out the triangulation, and this triangulation has a particular name called Delaunay triangulation. And this is very useful and used because mathematically this triangulation can be used for comparative modeling, like same geometric contract can be compared in geometric. Hence, that has been shown also by C, figure C, which is Voronoi diagram, and the dashed lines are actually shown in the C. These three figures actually is called to produce alpha shape, and that technique like alpha shape has been implemented in programs which I have described earlier to calculate the basic geometric modeling. This is a very efficient way of calculation of space left outside, and compare the space with a complementary space can be generated out of this. Deliberates how the pockets are identified. So once you know discrete flow of the system defined only by the empty triangles, and Delaunay triangles are also identified, which are not part of the dual complex. Then what one starts with the protein, makes the Delaunay triangulation representation, and then uses the convex hull, that is join them appropriately to deliver it or make out complex. Then you identify a few from the alpha shape concept, 5 obtuse angle, you can see them, and others which is not obtuse angle but the flow. So these flows, that is which are coming out of the obtuse angle, are shown in A and B bottom picture. So one are the obtuse angles, and the other ones is this, other than obtuse triangles, you have a flow, that is you come from 1, 2, 3, 4 and 5. Ultimately you go to the infinity as your triangle. That means it is the open space. So calculation or geometrical method is this, and it has been very efficiently coded in computers to keep this flow stored along with the alpha shape. So advantage of cast P is the pockets and cavities are identified analytically, and that's the best part of this surface program. And the boundary between the bulk solvent and the pocket, which is the active site lies, is precisely defined. Actually, this is important when you calculate the pose and accurately dock compounds. These are calculated parameters, all calculated parameters, and they're completely rotational invariant. So you can use, there is no use of any dot surface or grid surface, etc. to make here. Another added feature of using CASP is it can adjust the probe radius. That is, it is not necessary that you have to provide only one type of probe radius. It can adjust accordingly and simulate accordingly to identify the spaces. Actually talks about the pocket finder, another software which is server based. This is mostly energy based. This is the difference between CASP is that it is the energy based. So it finds the energy of the location by calculating the grid across the protein, and it also detects the binding pocket but also predicts the shape and size of the ligand which can bind. Tentatively it can bind. And this has been depicted in the picture on the top, which shows that if you use a probe of radius like rho shown here, then given the formula you can calculate the energy, and these energies are mostly calculated by grid potential by van der Waals forces. Generally only the van der Waals forces are calculated, not any others. You apply a threshold to eliminate the small pockets and just land up with the large pockets as it has been shown here. So here you not only get the shape and size, also possibility of energy, and the small pockets which are less than 100 angstrom cube are actually not counted. Advantages of this pocket finder is that it actually calculates and identifies the binding site location, even some of the times the receptors for which already no known information available for ligands, it can also find it, called orphan receptors. Not only that, it can actually, looking at the shape and size of the active site is given by this pocket finder, you can calculate and find out what are the ligands you may try here. So druggability of the pocket, that means how many other compounds can bind it. And then it has another advantage, that is you can calculate the potential of this, which is provided. You can also substitute the equations by electrostatics and get an estimation even sometimes. The only difficulty of pocket finder is that is very, very, very shallow pockets and the small pockets are omitted. This is most popular program called PASS. It's also a server. One can also download and run, but putative active site with spheres. Here we use only the probes. So taken a protein as shown in the picture, detail of this paper gives you that taken the protein, search for small probes. Here the probe size is important. Whichever clashes with the protein moiety is not taken, and goes on calculating all. this actually finds out even buried inside surfaces. As you see, he have been located not only whatever accessed sites on the bottom left hand side, the extreme, there is a circle collected which is buried active site. So that's one other important thing of this particular method. Actually talks about the representation of potential binding site, and that points ASP is a very common word that you can actually find, and many cases your output gives you that pictures. The probe sphere is important, and that is used here. So protein structures are filled with set of spheres, then filling the 3-point cannoli like sphere geometries are taken, then first layer calculated, then additional layers are calculated, and you go on finding the buried ones also. So we would like to continue that how the method is followed. So next is third filter. So what it done is no overlap with any protein atoms, then no clash with the protein atoms either, that is it should not be having inside gone into protein, and buried within the protein, which I mentioned. After that, you weed these numbers, because probes which are larger than 1 angstrom may be having a weeding necessary. Ultimately, you get the, by iterative systems, you get the existing layers. So first layer, second layer, like that you calculate and you get the full. Most often you should stop where the accretion phase, there is no new PS is found. Actually talks about the advantage of ASP. ASP techniques device very quick to calculate, number one, and then it can have multiple ASPs, and that's the important part. Computational of bound and unbound forms also you can calculate here, and it can identify many of the interfacial pockets, that is the between two other multimeric forms. So protein interaction might be also very advantageous to use these methods. Ligands only bind to the region which is enough buried and accommodate the volume. So that is commonly known to every one of the methods. Now I will discuss the every technique which has this APS technique, or what you call active site point finding by buried methods, advantage and disadvantage. Property based this kind can be also done, and coloring can be done, which is the advantage of this method. Also you can visualize these, because you can have a little bit time taken and color them with different kind of properties. And it also suggests many alternate binding site, that is somewhere else hiding. And these are major advantage, but there are few very important limitations, which is something like this. Now 3-point geometry, which is no identification of the flat cavities, so very much fails when you have a flat cavities. Also the described distances of these interaction sites, it may not identify for very small protein, that is the tiny bit proteins which may not be satisfactorily found its buried areas. Also the output and input files of comes out like a PDB format, and there is no information that what is the charge and how is the ligand and protein interactions are. However, this can be, limitations can be overruled or found out later on. Another probe-based method which is called LigSite, and this probe-based method basically captured the same grid principle. It uses a grid marked in the protein as it's shown here in the picture. Especially it shows where the atom position and then where is the these probes. So pocket identified by grid is shown by a small circle, and the big probing site is shown by a large circle. Also you can look at the directions. So the blue color shows not only the direction, that is the probe moves in whichever direction. If it hits the active site, it will come back. So basically the same Connolly algorithm basic theme is used, and this grid point is marked as surface if the vertex is within the 1.1 angstrom or so. All the rest are talked about is called solvent grid points, which identifies the pocket clusters and is called the surface solvent surface event. And Lixite scans all the 3 grid directions and 4 cubic directions, that is directional on the corners, and searches for the solvent surface events. So if solvent grid point is a part of at least 5 surface solvent surveys, it marked as pocket. So minimum 5 space should be available. Or if all the pocket grids like this collected accordingly, and then you can see the large grid clusters, and that is the new cluster coming out. The top 3 clusters are always retained and re-ranked according to their conservations involved in their surface residues. Even sometimes the list of residues are also mentioned here. SurfNet. As I told in the beginning, SurfNet is a program which also takes care of the surface probes. However, there is a big difference between the Lixite and Surf. SurfNet also available online and hosted by the UCL, but College of London. But here it generates surface area and the gaps between the surface area of the 3D from the 3D coordinate. The gap region corresponds the void and void between 2 molecules, and it can calculate the cleft, the cavities, and especially it can calculate the binding site, not only simple binding site, side chain interactions, but in addition it can show you the plane, paraboloids or geometrical figures, which it does. Here I elaborate mostly in the slides how the surface are stored, and SurfNet calculates the density also and contour by contour level of 100 on 0.0 level. So here you represent the protein atoms which are by circles, open circles. Each pair of atoms you can see the striped ones. Test sphere, which is in gray and circle is dotted, are created between them. Here you do not have a simply 1.4 or water probe size as a size. Here we are using, SurfNet uses variable radius probe, which makes it very efficient and accurate. So it fills up with a variable probe every interaction site. The left hand side picture demonstrates one such point, and right hand side when it filled up the whole process until it spaces no obstruction. Shows you elaborated form how to fill the gap between the atoms, because this is most crucial point of calculation of SurfNet, which brings out the accuracy. It's pictorially said from the top pictures and bottom panels. So first you see the gap regions between 2 molecules. Then each gap sphere is placed between pair of atoms, which is shown between a straight line connecting them. And then the gap sphere is placed right in the middle of that point. That is, center is dissected by the straight line which is connecting it. And the size of the sphere is determined when it hits the both pairs. Third picture shows that van der Waals touching each other. Then the picture D and E continuously midway of the van der Waals spheres just touching each other, and radius of falls below some predetermined minimum limit, normally 1 angstrom, because any 2 atoms cannot go, even it is hydrogen, cannot go lower than that. So it is a very conservative, it is rather internal, much more goes into it. Finally, figure F shows when all of these spheres are continuously done and filled up the whole gap region. It stops only when you find another coordinate of the sphere which is superimposing the same place. However, later on you remove the gaps, but you get the circular spheres all the way. Here we discuss about this program and its advantages. The most important here is that you can actually find density values of this. Or that means you can look into the 3D grid of density values using a Gaussian system. So which means on the x-axis, if you look at, is the distance from the points, and how much densely the system is packed is plotted on the left hand side, y-axis across. Now, the grid points within 3 times of the radius of the atoms are updated as the density value become negligible after the 3 times, which is shown in this figure. using this, the cavity and the pocket detection algorithm, this is very fast. Not only it is very fast, it is also connected to the graphics program and can have even the crude amount of gap regions. So one can calculate how much we are losing out when you are actually measuring the whole active site space. In the list of all the methods, I've covered the geometric methods and the probe methods. I said that we should address this evolutionary method. It is basically to take advantage of already known information which evolution has provided to us and then go on working upon it. Now, this method is called concept methods and it's been hosted also from Illinois. But you can have a look at the whole site, which I am just giving you a short summary of the method. Basically, it takes many of the similar and collects alignment done. Once the calculate the conservation score of this alignment, empirical Bayesian or maximum likelihood method is applied, so that we get a MSA, which is alignment method, gives us like a phylogenetic tree preparation. These proteins with the conservation scores first colored on its surface, and it can be even visualized by programs available on JMOL, so that the first it gets the conserved amine and then calculate the surface area for them. And that gives you the picture of surface area, colored picture of surface area, which are exposed all across the evolutionary programs. Now, I would go to the next slide. Before that, I would like to just emphasize one thing. These are different methods, and I chose to describe these methods in detail so that the basic theme of these methods can be understood well. It depends on what type of protein one handles to decide which methods I should be using. However, next slide contains whole detail of the methods, comparison, faster, slower, whatever, etc., etc. But one must understand that the methods used can give different level of accuracy at different level of proteins, surface, finding algorithm as you would like to use. It's according to your necessity, you need to choose these methods. Here I show you a comparison table which also will be in your text and you can read it from the paper cited earlier. Most important is to note down that some softwares do very quick but not so accurate predictions, like fastest way of predicting pockets are Pocket Finder, CAST-P and Subnet. However, PASS is a bit of a slow, however, it has some advantage. And what it does is, it can find small buried files, that is small buried pockets, which none of the others can find. QSite, on the other hand, is not so fast. However, that is been found to be more accurately find the active site or binding site, which are experimentally has been found out. And the Pocket Finder, however, is used by more cases and along with PASS, which predicts shallow and small molecule binding additional active sites. So this has to be remembered when you use a protein. However, one should advisable to use all the methods and then choose whichever you would like to utilize for your docking, depending on what kind of compounds database you have with you. Next slide summarizes about the whole of references. Hence, I would like to get this list also will be available in your write-up. Now I talk about the summary slide. This active site detection plays the most important role in actually accurately how you predict the binding efficiency. And I have discussed here three major different methods and to find the active site, and their advantage and limitation also has been discussed. What we notice here is geometrical methods actually coupled with the rolling probe probably have the best advantage to map the active site of the protein. However, buried active sites sometimes may not be possible to find if you do not use exact program to find them. This is the first step to proceed docking. Hence, your docking accuracy, etc., very much dependent on this particular step of identification of active site. In addition, some of the methods can actually give you chemical interaction map as well, so that your docking and posing of the molecule properly can be achieved at the end. Thank you.